All right, we'll just get started as usual with some updates, announcements. I will just give a quick legislative update, which is that our hearing in the Minnesota House for the two bills related to Forever Green was delayed last week thanks to the snowstorm. Kind of unprecedented. They shut down the legislature for a few days, and so ours got bumped, but luckily they turned right around and rescheduled it for this coming Tuesday. So very short delay, no issues. In fact, made my life a little bit easier. Last week was extra busy, and it would have been hard to get my hearing testimony together. So now getting ready for Tuesday, we've got a great slate of testifiers. We have both bills being heard, and we're all set with the authors. We met with them last week too. So looking forward to a good hearing. And like I said in my email, if you're interested in joining, let me know, and I'll try to pull together a carpool from campus. That's all I have as far as announcements. Anybody else want to share anything before George gets started? Going once, going twice. Okay, George, go for it.
Okay, thank you, Mitch. So today we have some updates from what we've been doing in the Food Science and Nutrition Department. So this update is from myself and from Pam, and it's mainly on cancer. So you can see now we have a very nice introductory slide. So I'm told we all have to use this now. And one important thing is to make sure you let us know your pronouns so we don't call you a different pronoun. Okay, all right. So I'll be talking about 3 projects that we've been working on here on cancer. I will start with one that has to do with the brand. So we're looking at exploring steam explosion to stabilize intermediate wheatgrass brand. And this is done by my PhD student, Radhika, who is currently in Norway doing this research. So why do we have to do this? Why is this important? So, I mean, we do make bran when we refine cereal grains, and particularly for intermediate wheatgrass. Radhika has been working on developing tempering conditions that will allow us to effectively produce refined flour from intermediate wheatgrass. Now, with the current state of intermediate wheatgrass, we have about 40% bran after we do the refinement. So the question is, what do we do with all that bran? I mean, there's that option of having the whole grains and using it as a whole flour. But as we all know, in most cases, the refined flours do much better for baking applications than the whole flour. So we have 40% of bran. That is a lot of bran to discard. So Radhika and I decided to do something about this. And it is also based on the fact that the bran itself is a nutrient-dense co-product of milling. And there is definitely something we can do about it. So bran is rich in health-promoting compounds. We have the phenolics packed in there. We have some lipids also in there. And then it also serves as dietary fiber. But because it has a lot of lipids and also has a lot of lipases or lipid-degrading enzymes, the use of bran can sometimes be problematic because of shelf life. You can easily have rancidity going on because of the lipid-degrading enzymes. So we came up with this idea of steam explosion, and I didn't have the equipment here. And luckily, we have a colleague who used to be in this department who is now at the Norwegian University of Life Sciences, Katrin Tell. So she has the steam explosion equipment, so we decided to give it a go. So this is how the equipment looks like. It's pretty much similar to the puffing that I'm doing, but this here uses really high pressures. I mean, we are talking about from 100 to 700 PSI, really intense pressures. So you load your sample in there, and this is the feeding chamber. This is where the reaction goes on. So your sample stays in here, and then it is exposed to heat to about 260 degrees Celsius, and also very intense pressures. Treatment time can vary. I mean, you can do as minimum as 30 seconds all the way to 20 minutes. And then when you reach the time of treatment, the pressure is released instantaneously. So pretty much like how we do puffing. So why do we do this? Steam explosion has been found to improve the nutritional content of products that are steam exploded. In the case of bran, we would be able to first inactivate enzymes, the lipases and all the enzymes that are in the bran that may cause issues can be inactivated. Therefore, you can extend shelf life. There's also research that has shown that when you do steam explosion, you can increase protein solubility. You can also increase the amount of soluble fibers in there. So we wanted to see how intermediate wheatgrass bran will do when we do the steam explosion. So this is the plan for the research. We plan to do treat the grains, the bran with the steam for 130, 150, and 170 degrees Celsius. And we want to go to 10 minutes. I mean, we could go higher, but we wanted to see what would happen when we do 5 and 10 minutes. So after the steam explosion, the samples will be collected and then they'll be freeze dried, they'll be milled, and then we do all the physicochemical analysis. So these are some of the analysis that we've planned to do. We want to see the effect of steam explosion on the lipid degrading enzymes. We want to see the effect on these other physical characteristics. So the color, will it change the color? Will it impact any residual starch that we have in there? What about other enzymes, so polyphenol oxidase? And what about hydration properties, so swelling capacity, water binding capacity? Are we able to improve this or change this when we do the steam explosion? Remember, these hydration properties are very important when it comes to baking applications as well. And then we look at free fatty acids, peroxide value, and other components that may actually change because of the steam explosion. And we also look at antioxidant content of the samples after they are steam exploded. So some preliminary results that we have so far seems to suggest that when at the initial steam explosion treatment, we seem to have a reduction in free fatty acids. But when we hit the 150 psi for 10 minutes, it starts increasing. And when we treated for 10 minutes at 170 psi, we got the highest amount of free fatty acids. When we hear free fatty acids in your sample, you always have to worry about stability or shelf life, because these free fatty acids can be oxidized and then you get a lot of rancidity. That is there when you are looking at oxidative rancidity. At least we know how we can prevent oxidative rancidity. What we were worried about were the enzymes, the lipases, the lipoxygenases, because these enzymes are the ones that will start hydrolyzing these fatty acids, the lipids into fatty acids. The good thing is that even though we had high amounts of fatty acids, we did not have any activity for the lipase or lipoxygenase at that when we did the steam explosion, which is good. So it means that these high free fatty acids that we see in here are actually coming from the process itself and not because of the enzyme. When we looked at color, we just looked at the brightness. Of course, the more we did the longer we did the steam explosion and the more we increased the temperature, we saw that the samples began to become darker and darker. I mean, I think this is expected, but we are yet to see how this reduction in lightness would impact products when we actually use them for. But yeah, steam explosion resulted in a reduction in the lightness. And then protein solubility, we saw a huge increase in protein solubility at the 170 psi for 10 minutes. Increase in protein solubility is really significant in that when you take the bran, we pretty much will see it's pretty much made up of fiber, and the proteins in there might not be very soluble, as you can see from the slide here. This is for the control. Steam explosion resulted in a gradual increase in protein solubility. If the proteins are soluble, of course, it is more available for the body to use. So steam explosion resulted in a significant increase in protein solubility, which I think is really good for any food uses of this product. We also looked at antioxidant, we also looked at the phenolic content, and we can see again, as we do the steam explosion, we see a lot more release of these phenolic compounds. And it makes sense. Most of these phenolic compounds are bound to the cell walls and the cell wall components. So when you do the steam explosion, you are basically releasing these phenolic compounds. And if you release these phenolic compounds, chances are that your antioxidant capacity would really be high, which again is something that is very desirable in product. So by doing this steam explosion experiments, so far we've seen that this has the ability to really improve the nutritional profile of the brand. We are yet to measure the hydration properties and all that, and hopefully we can also change the hydration properties for the better. So we can now add these steam exploded brand products to bread or to baked product or to other products in a bit to increase the fiber content. So that's the kind of the preliminary results that I have for this study. And this, as I said, is being done in Norway with a lot of support from the university. So we leverage a lot of funding from the university. This was not directly funded by Forever Green, but part of the work is going to be done here after the trip from Norway. So yeah, this is, I think it's a very interesting study. The second study that I'm going to be presenting on is from Leslie, who is a master's student with Pam. And Pam couldn't be here today and to do the presentation. So I'll try my best to present this, but basically looking at storage stability of intermediate wheatgrass flour as impacted by agronomic practices, breeding, and commercial germination and extrusion techniques. We've been doing this study on storage for quite a while, and so far it seems like intermediate wheatgrass stores pretty well. Some components do change, and I'll talk about some of them, but we've been getting a lot of requests from organizations in the EU. I think there was 1 or 2 requests where they were pretty much interested in how well intermediate wheatgrass stores. So this study sort of gives us an idea of how well intermediate wheatgrass stores. So this study is broken into 2 parts. There's a first part of the study where we're doing a lot of pre-screening, and the idea here is to analyze a lot of varieties and Minnesota Clearwater varieties grown in different locations, and to look at the impact of breeding and the agronomic practices that are being carried on with these grains. And then the second part of the study is where we are looking at the storage. So we want to track changes over storage, and we selected 3 varieties of interest. And among these varieties, we extruded some of them, and we also germinated some of them. So we wanted to see whether these processes improves or reduces shelf life. So the experimental design, we had a lot of samples, and I'll show the samples when I get into the results part, but these samples were pre-screened for proximate composition, carbohydrate characterization, antioxidant content, and then enzyme activity. And then after that first pre-screening, we selected a few samples, and I'll explain the samples we selected and why. So we selected a few samples, and then we extruded them and germinated them and pretty much pre-screened them again. Then we took these samples and then also stored them over a period of time. I think we went all the way to like 8 months. And then over that storage period, we kind of monitored some of these chemical changes in the products. So these are the analysis that we did. It's a lot of analysis, but I won't present all of them. I'll present just a few of them today. So for the pre-screening data, this is a table that shows that. Now we had a lot of MN 1601, 1603. So these are some of the varieties, different varieties that we got from the breeders. And then we also had MN Clearwater that were grown in different parts. So we have in different parts of Minnesota, and we had one that came from Kansas. Now, when we look at the protein profile of these samples, now within these varieties, we see that there are for significant differences. The protein, the MN1601 had just 16%, all the way to 21.1% for the 1802. That's a big difference. Now, if you compare these to the one coming from Kansas, the Clearwater that was grown in Kansas had the highest protein content. Environmental factors could be due to maybe elevated temperatures in Kansas compared to Minnesota, or maybe the soil in Kansas being better than what we have here. But we can see clear differences from the locations as well. When it comes to fat content, we saw some differences as well. The one from Kansas had 4.87, but MN 1801 had 4.33, 1603 had 4.13. On average, the fat content is about 5%, and that is still very high. And that is always a worry for us as food scientists when we look at the shelf life of intermediate wheatgrass. Not only does it have very high fat content, but it has also high lipase and lipoxygenase activity. So those are pretty much what you wouldn't want to have in a product if you are concerned about lipid oxidation. Ash content around 2%, saw some slight differences within the varieties and also due to the location. Total carbohydrate, total starch, total carbohydrate was pretty very similar for all of them. A little bit of a difference in total starch, and the one from the Clearwater variety from Kansas had very low starch content, and also the one MN1603. When it comes to amylose and amylopectin, they were pretty similar. We also found very similar differences in the dietary fiber as well. So the big difference that we found was mainly in the protein content, and for us it's a huge thing because the protein content contributes significantly. Well, not just the content, but the content contributes significantly to the baking characteristics of intermediate wheatgrass. This here shows the SDS page, pretty much looking at the molecular weights of the different protein components. So here we have our standard reference standards with known molecular weight. This here, the number 2 is the one for hard red wheat, and all these are the different varieties of intermediate wheatgrass. We can clearly see that intermediate wheatgrass significantly lacks the high molecular weight glutenate. So we see that clearly shown in the wheat sample, but intermediate wheatgrass doesn't have that much. And we've already seen the impact of this for baking applications. Even though intermediate wheatgrass have both gliadins and glutenins, it doesn't bake very good bread when you compare it to wheat. And it's pretty much because of the absence of these high molecular weight glutenins. And we found that in pretty much all the varieties that we studied. It wasn't very clear whether there were significant differences between this, but visually you can see they really lack these high molecular weight glutenins. This here is also some results on polyphenols. So ferulic acid was the one that stood out. Between the samples, intermediate wheatgrass samples, there were some differences, but we clearly see that when you compare all the intermediate wheatgrass samples with that of wheat, intermediate wheatgrass samples have pretty high amount of ferulic acid. The same with synapic acid. When it comes to p-cumaric acid, there are some intermediate wheatgrass varieties that have lower amounts when you compare it to wheat. Now, having high amount of ferulic acid can be good because then it confers more antioxidant property, but ferulic acid is also known for astringency in products. So I personally have not really picked up any astringency in intermediate wheatgrass product, but it is something that we would have to keep an eye on. When we look at the antioxidant capacity, we see that DPPH for hard red wheat is 11.2, and all the ones from intermediate wheatgrass are pretty very high, and it's all coming from the high phenolic compounds, so which is good. When it comes to lipase activity, intermediate wheatgrass, we saw variations in lipase activity between the varieties, with the same thing for lipoxygenase activity within the varieties. But again, if you compare all of this to hard red wheat, you can see that significantly high lipase activity in intermediate wheatgrass, which is a huge concern because it also has a lot of fats. So we need to come up with processes that would be able to inactivate these enzymes so as to reduce the amount of lipid oxidation that we get in these products that are made from intermediate wheatgrass. I mean, 0.3 to around 3.05 or 4.11 is huge. So the pre-screening, what we got from the pre-screening was that intermediate wheatgrass compared to wheat is still very high nutritional, have very good nutritional profile. And we see that this detailed compositional data set that we are generating or have generated would actually help in the development of new traits in new cultivars that are being developed. We hope that we can start trying to breed for intermediate wheatgrass that has low lipase and lipoxygenase activity. Looking at the results that we've seen so far, I think hopefully we hope that this will be kind of like a priority answer point to see whether we can reduce the amount of lipase activity and lipoxygenase activity. Not so much of the fat, but if we can reduce some of the fat, that will also be good. So for the storage study, what we did here was we selected 3 varieties. So we had one Minnesota Clearwater that came to us from Ari, and then we had 1603, and then we had 1601. And this selection was based on the amount of fat we have in there, the amount of antioxidant we have in the samples, and also the enzyme activity. We wanted to go to the extreme. We wanted samples that have the most fat and also the most enzyme activity, which is the lipases and the lipoxygenases, and also look at samples that have very high antioxidant activity. And then we want to store them and see how well they fare over a period of, say, 8 months or a year. So the samples were milled and they were put in these very nice containers, and they were incubated at room temperature in a humidity chamber. And with this storage study at a relative humidity of 43%, which is pretty much what you find in most homes or in most factories. Okay, I think it's frozen. Okay, all right. Okay. And then we also had some of the samples extruded. So before we did the storage study, we extruded some of the samples. So we used a wheel screw extruder, and then these were the conditions that were used for extrusion. And this here is a picture of the extruded product. So we wanted to see whether when we extrude these samples and store them, they would store much better than the regular grain if it's not extruded. So we use screw speed of 200 RPM, and these parameters were optimized in a previous study from a student. We use feed moisture of 24%, so we had to condition the grains to 24% moisture, and we use a feed rate of 2 kilograms per hour. And then these products came out through a die, a 3mm circular die, and this is how they look like. So after we did extrusion, these samples were dried. When they come out of the extruder, they are pretty dry, but still has some amount of moisture in there. So we dry them overnight at 50 degrees Celsius, and then they were stored at refrigerated temperatures before the study began. We also did some germination, and we steeped the grains for 3 days to get to a moisture content of 43 to 45%. And this is how the grains look like after we steep them. So during the steeping, we had to change the water every day, and then they were aerated every 15 minutes. And then after the 3 days, we drained the grains, and then we germinated them at 16 degrees for 4 days. So this unit here is from Sprout Labs. You are able to keep the temperature. It has a cooling unit in there and also has a heater in there. So it was able to keep the temperature at 16 degrees. If you don't keep it at 16 degrees, the grains will generate so much heat that either your germination will be so fast or you pretty much will kill the grains. So during the germination, they were stirred every 8 hours. It is important you do that, because if you don't do that, the roots get entangled into each other, and it's very difficult to separate them. And the stirring was also done to prevent any mold growth. The conditions that we were using was very suitable for mold growth. So the same thing as we did for the extruder, after the grains were germinated, we dried them or kiln them for 2 to 3 days, and the target moisture was 8%. So for these samples, this is the test that we did. So we stored them, and we had the control samples. These are the untreated grains, and we stored them for 8 months on average. So we took samples at month 1, month 3, month 6, and month 8, and then we looked at how all these parameters change over time. So I'll present some of the results, not all of them. So this slide here shows the kind of like the proximate composition and also some parameters on starch on these samples. So we have Minnesota clear water, we have the untreated, extruded, germinated. We have the same for 1603, untreated, extruded, germinated. And we couldn't germinate and extrude the 1601, but we still included the results anyway. If you look at the results, the protein, the fat, the total carbohydrate, you could see that there wasn't much of a change in the product when they were extruded, and also most of it when they were germinated. It is good in a sense, because we don't want these processes, like we don't want to extrude the grain and significantly reduce all the protein or significantly reduce all the carbohydrate. So that was good for us. At least the protein remained very consistent. We see that in pretty much all the varieties. When it comes to total starch, we had some slight decreases in total starch when we germinated the grains. And this is expected because, of course, the enzymes that we activate during the germination are using up these starches and breaking them into soluble sugars. So we saw that in both varieties. This is not a bad thing, it's just converting them to soluble sugars. When we look at dietary fiber, when we look at the soluble dietary fiber, we see that when we extruded, when we did extrusion, there was some reduction in the soluble dietary fiber. We also see that in the germination, and this trend was a little different for the MN 1603. So when we germinated, when we extruded, we saw an slight increase in the soluble dietary fiber. And the same was seen when we germinated as well. I mean, increases in the soluble dietary fiber is good, and the body makes use of the soluble dietary fiber better than the insoluble dietary fiber. So typically we want to see products that have a little bit more of the dietary fiber. But if you look at the absolute values, they were not really very high. So we can conclude from these analysis that the germination and the extrusion did not significantly change these grains, which I think is a good thing. Here we look at the SDS page of the germinated, the extruded, and the ungerminated samples, same gliadins and glutenins. And what we see here is number 3 here represent Minnesota clear water that is extruded. Number 6 here represent the MN1603 that was extruded. And you can clearly see that the intensity of the proteins, especially the high molecular weight proteins, are pretty very faint. And this is probably because that high pressure and temperature that we use to extrude is pretty much breaking down or depolymerizing these high molecular weight proteins. This could mean that there could be an increase in the amount of soluble protein, which is again very good because we want the proteins to be much more available when we consume them. So that is what we found. Germination did not really have much of an effect on the protein content, but extrusion really did. During storage, we saw a significant reduction in carotenoids. So we saw lutein and zeaxanthin kept going down over time. And intermediate wheatgrass compared to wheat is a much significant source of carotenoids. In fact, intermediate wheatgrass has so much carotenoids that the refined flour actually has a yellowish taste to it. But the bad thing is that as we store them, we lose the amount of carotenoids. So that wasn't a good thing for us. So we would have to come up with processes that will probably slow down how much of these carotenoids are being destroyed. So much of it can actually be slowed down by how it is packaged and whether we have oxygen or whether it is exposed to light. So that was something we saw in there. Free fatty acids, and we saw them go up. Free fatty acids was going up for pretty much most of the samples. Peroxide value was also going up, but I don't think we saw that in the germinated and the extruded sample. So that was also not something we wanted to see, but yeah, it actually happened. And so extrusion inactivated most of the lipases and it reduces lipoxygenase activity, which is good. Germination did increase enzyme activity as expected. Free fatty acid content increased and then kind of stagnated, but that increase could result in significant problems down the line when it comes to shelf life. Peroxide value stagnated, but increased after 3 to 6 months. So, and carotenoid content, as I showed, decreased significantly, which to me is a concern. And so the extrusion, we found that it has a potential that of producing varieties that are much more shelf stable. And we've been working on extruding intermediate wheatgrass in the department. We've been able to optimize the conditions for extrusion. We are hoping to kind of scale up the process, but extrusion was found to be one of the processes that can be used to significantly increase the shelf life of intermediate wheatgrass. The other study that we've been working on is investigating the functionality and use characteristics and processing specifications of intermediate wheatgrass grains across stand life and crop varieties. And here the objectives are to evaluate and compare the chemical composition and protein profile of intermediate wheatgrass harvested in 3 subsequent years. So we know yield goes down when you harvest these grains, but in addition to the yield going down, does it significantly change the grain? I think this is a very important thing that we need to know, because this would help us kind of advise the processes to probably change some of their processing parameters. What we hope to see is not to have a significant change in the grain, but we'll see at the end of the study. And then the second objective is to investigate and compare the functionality of the harvested grain. So not only are we interested in the chemical composition, the protein, the carbohydrate, but how well does it bake bread, for instance, the functionality of that gluten. And then a third objective is to work with the industry and to pretty much to work with the intermediate wheatgrass industry and partners to conduct applied research on production, seed cleaning, dehulling, milling, sifting. We have all these processes that we already have to some extent, but what if the grains change year on year? So we need to work with our industry partners to figure out how we will respond to this problem if it indeed happens. So that is the third part of this project. So far with this project, we haven't started the actual analysis, but we have samples already received. Samples harvested in 2021 and 2022, MN 1902 and 1904, and we already have these samples. So they will be harvested again in 2023, so we can get all the 3 years. 1602 and will also be grown and harvested for the third part of the work. This is where we are looking at working with industry partners. And data from the 2021 and 2022 shows that 1603 is doing well. This is what I got from the breeders, so that's good. We have some small quantities of 1901 and 1903 and 1905, which may also be part of this study, but hopefully we will start doing, we already have a student who is preparing to start analyzing the 1902 and 1904 that we've already received. We've also been doing some work on puffing and germination, and I have a master student, Dana, who is working on the puffing and the germination. We started working on the puffing. Most of you have seen that, I've seen some of the puff grains, but the samples that you guys have seen so far was just done preliminary. So there was no like design of experiment to it. So this time we want to really understand how the puffing pressure and the moisture does affect the grain, right? So Dana is working on that. This is our experimental design for the puffing. So we want to look at moisture content of 10, 15, and 20%. And also the pressure, the puffing pressure, we're looking at 100, 130, and 160. So we want to pretty much look at the effect of the moisture and the effect of the pressure on the way the grains puff. We've done some preliminary puffing, and here you can see this is all grains that were puffed at 10%, right? So you can see that even at 100 PSI, the grains do not puff. We went all the way to 130 PSI. Then a few of them started puffing, but if you should ask me, I would say this is still no puffing. When we hit 160 PSI, we've got the grains to puff. Now, what you can see here is that as we increase the PSI, we start seeing a little bit more browning. And not just do we see browning, I can actually smell some burnt flavor to it. So these are some of the things that we'll be digging into to figure out why this is happening. So this is 10%. We also did 15%. So 15%, even though we increased the moisture content by 5%, 100 PSI still did not puff. 130 PSI, we also got some puffing, and it looked like the 15%, 130 PSI got a little bit more puffed compared to the 10%. And then again, when we hit 160 PSI, we got the puffing actually happening. So if you compare the 10% 160 PSI and the 15% 160 PSI, we saw that the grains, the puff grains from the 160 the 15% 160 PSI were much bigger. In fact, when we placed them in the containers that we were using, we saw they had significantly expanded compared to when we used 10%. So what we've seen so far is that, yes, there is an impact from the pressure, and there's also an impact from the moisture, but the impact from the pressure seems to be much more significant. And of course, as we increase the moisture, we tend to see a slight increase in the expansion of the grain. We also saw that at the 15% moisture, we did that burnt flavor, intensity of that burnt flavor actually subsided. At 15%, you could now smell more of a cooked grain rather than a burnt grain. So that was good to know. We will be doing the same experiment with a 20% moisture. We were supposed to do it this week, but then the snowstorm prevented us from doing it. So we'll be doing a 20% moisture to see how it impacts the expansion and also it impacts the flavor. Not only are we interested in the expansion and the flavor, but we want to see whether this process is changing the grain. So even if we didn't have it puffed like these ones, does it change the grain in any way? So we are going to melt this into flour and dig into the nutritional characteristics and also dig into the functional characteristics as well. The germination work we haven't started yet because we had some issues with our germination unit, but we'll also be doing a similar experiment. We'll be looking at germination time and also the temperature of germination. So that is what I have for today. So if there's any questions for me, I'll be very happy to answer.
George, could you give the group an update on the big project in New Zealand?
Oh, so the big project in New Zealand, they sent the first report where they saw,
Give them the background. Give them the background.
Okay. I probably should have added it. So we have a project with a university in New Zealand where we are looking at the consumption. It has to do more with the consumption of intermediate wheatgrass and whether when you consume intermediate wheatgrass, it has any changes in your, like how you get hungry, right? So they have been working on this. We started this like, I think last year, they did the first part of the study and I forgot to add the report to it, to this. And I don't remember the details in the report, but they are doing the second part of the study where they are still continuing to validate what they found in the first experiment they did. I can share the initial report that is sent to the group for people to know what we are doing on that.
They're actually feeding it to mice, right, George?
Yes, yeah. So they are feeding it to, it's more nutrition. So we do a lot of the processing and they are doing more of the feeding. When we feed it to mice, does it give the same satiety? Does it give the same like response when say you eat another grain? So that's what they are doing. So it's more nutrition related. Yeah.
Cool.
They're also doing evaluating glycemic index, right?
Yes. Yeah. And for glycemic index, so they are using rats and we do some of the lab scale glycemic index, the in vitro once here. We do that here. We'll be doing it on the puff grains and the germinated grains as well. Yeah. So there's a question here, I think from Mitch, is fiber supplement the most likely end product? Oh, you mean for the steam exploded? Yeah, so fiber supplementation could be one of the things that we'll do, but we also want to see whether if we add it to say, so intermediate wheatgrass still has an issue with bread baking, is if we add that steam exploded fiber back into the refined flour or even into wheat flour, does it make it any better? But yes, it will help to increase the amount of fiber that we have in product that we add it to. And then there's a question here regarding reducing lipase. How feasible is it to run this analysis on few hundred samples or few hundred space samples? How many grams are needed? How long does it take? Yeah, well, it doesn't, I would say it takes too long and we can definitely run some few hundreds of samples. I know when breeders say few hundreds of samples, they are looking at 200 samples. So, but yeah, it's something that we can run just like we run all the other analysis. It doesn't take too long to run that. And then, do you have the steam explosion device? No, we don't have it here at the UMN. And so that's why the student had to go all the way to Norway to do it. So we have a very good links with the university in Norway, so we can still continue to do more experiment on steam explosion.
Explain to the group how you have that link to the university in Norway.
So we had, we used to have a postdoc here who is called Catherine Tell, and she worked a lot on intermediate wheatgrass when she was here. She moved to, she got an assistant professor position at the University of Georgia, and then later on moved to the university in Norway. And since she moved there, we've still been working on intermediate wheatgrass, and she actually sometimes do join these meetings. So she reached out to my student Radhika, and we kind of were thinking about what experiments we can do with her. And then she came up with this idea of, oh, we have this steam explosion equipment. And so we said, yeah, let's give it a try and see how it'll work. So Radhika went there. Radhika traveled to Norway I think in November last year, and she's been there. She'll be back in March. So yeah, that's how we got to do it there.
Well, all I'm saying is it's a good example of how the investment in the Forever Green, not only has local, but obviously it has international connections that are built off of the work here.
Yeah. So we are in Norway, we are in New Zealand. Yeah, we are expanding.
George, there was a second part to Bohu's question. He's asking about, does the steam explosion generate furfural or HMF? And are they okay in food?
We haven't looked into that. Furfurals and HMFs are always a little bit of an issue in foods, but we haven't looked at those yet. It is something that we can measure. Yeah. But yeah, those high temperatures and high pressures are likely to generate some of these furfurals and HMFs. Yeah.
And I had another question picking up on Praveen's question of, can we screen our breeding stock for some of these key characteristics? So in addition to lipase levels, what else would you identify, George, as some critical targets there?
Yeah, I think the lipase, we can definitely add them to the screening we do. So lipase is something that is important, and also the high molecular weight glutenins that I talked about, intermediate wheatgrass is lacking in them. So if we can have a way to screen a lot of the samples to be able to identify the genes that control these, that will be very helpful. I mean, if we can increase those high molecular weight glutenins, that will significantly improve the baking properties of intermediate wheatgrass. Yeah, because when you say intermediate wheatgrass, the fact that you have wheat, people always think, oh, it's similar to wheat. But then when you bake the bread, it's like, okay, yeah. So those will be the two things that I would be interested in screening for.
Gotcha. Thank you.
Any more questions?
Yep, I have one.
Okay.
It's more of a comment than necessarily a question. I noticed that you are just focusing on University of Minnesota currents of germplasm. As you well know, there's breeding programs in Canada, and obviously our big partner, Lee DeHaan at the Land Institute, is releasing material. All I would suggest is that I would encourage you to make contact at least with Lee DeHaan. Because that germplasm out of that program, will have utilization across the country and even in Minnesota. It would be nice to have a broad array of germplasm in your food science research.
Okay.
I would encourage you to work with Lee or talk to Lee or through me or others to sit down and have a Zoom conversation about the possibility of availability of some of that germplasm that could be brought into your food science program.
Okay, I think that is a good idea. And I do remember we had a discussion like this before in one of the meetings, and we were not sure whether we could use these funds, which are like for Forever Green Estate funds, for analyzing samples out of state. So that was something that came, I don't remember what meeting that was, but
No, I don't think that that's an issue. We can check
Okay.
with Jeff, but I think the only issue is that the money is spent in Minnesota.
Okay,
And from a scientific perspective, it would be kind of silly, right?
yep,
Not to have,
To
not
have
to
other
use our
varieties, yeah.
funds on looking, across the scientific question. Because some of that germplasm is going to be grown in Minnesota as well, right? So Jeff can, if he's still on, I don't think he would suggest that there would be any issue
Okay,
of evaluating material from no matter where the hell it is in the
Okay.
world, if it has a connection to Kernza development.
okay, yeah. I mean, I don't think it will be a problem for us to analyze those samples. So yeah, if we can meet with,
No, I'm just not saying you're doing, but I think that we ought to think about having a platform that or a program that basically includes other germplasm from across the country.
okay, yeah. I mean, we're doing something, I think under the Kernza Cup, we have some varieties coming from Wisconsin and other places too. So yeah.
Yeah, I just think there ought to be a more comprehensive framing of the evaluation around this issue of just the germplasm and where the production is at. You already had some material in your studies from Kansas,
yeah.
right? But I would just suggest that we have a team think that through.
Okay.
In terms of the variability that's coming out of the various breeding programs, and how that variability might impact the food industry.
Okay. Okay.
Peter has his hand up.
Yes.
Thanks, Don, and thanks, George. This is really interesting stuff. Not a scientific question per se, but I'm wondering if you have a sort of ballpark figure in your head for how long it might take until most of these kinks are ironed out in Kernza, and it's sort of, obviously it's going to be constantly improving, but to the point where the food industry feels it's sort of widely usable.
Mm-hmm. I think as Kernza is now, I think it is widely usable. I don't think there's any issues for it not being used. From the shelf life studies that we did, what we did here was like accelerated shelf life studies that we did, but it seems to store very well. We've had grains that we use. When I started here, we had some grains that we did analyze, and apparently those grains were grown like 5-6 years ago, and we had no idea. And it's still big bread, it still did very well. I always tell people, as far as you store the grains, just like you store wheat or corn or any other grain in a dry environment, just like you store any grain, Kernza should store very, very well. Hopefully, we should be able to, most of these experiments that we are doing, especially the one that is important to me is the one on the stand life, the one that we're looking at the different years. That I think is very important, and hopefully in the next 3 years, by the time the research is done, we should be able to have a very good idea as to whether the grains change. Because if it does change, then we have to think about how to blend. How do we blend the grains from the different years to keep that quality still consistent? And a significant part of that project is working directly with the food industry partners to figure out what to do if we see those changes. So I don't think it is far off. We should get most of these things sorted out pretty soon.
Thanks.
I see Lee DeHaan is actually online. Lee,
I'm here.
wake up. Hi, Lee. Hey, Lee, what are you doing in terms of your germplasm? Do you have any food science partners that's looking at your germplasm?
Not specifically, no.
Yeah.
It was interesting to see from the data here. It looked like the location the stuff is grown is probably more of a driver than the germplasm itself.
Yeah, that's right.
That wouldn't surprise me. So for that reason, usually don't expect things to change that quickly on
Mhm.
the genetic side. More it's on where it's growing. But
Yeah,
we do have I've followed up with what was started at the University of Minnesota on breeding for baking and mixing quality with the North Dakota State USDA program there. I've done several generations of selection now, and this past summer grew enough material to actually probably bake some loaves of bread with it. I think I've maybe sent an email to George about it. We
Mhm.
might looking for someone to evaluate whether that product that really worked or not.
Mhm.
Yeah, we were selecting for basically the high molecular weight glutenin content.
Yeah. Yeah. I mean,
How-
yeah, we should be able to work on that with you.
what is the status? Were you able to increase it dramatically?
That's what I don't really know necessarily.
Yeah.
So every year we would evaluate individual plants and quantify it. And yeah, it looked like it was going up in those plants, but whether it turned out to something that was actually, we wanted to do several generations to go far enough that we could
Yeah.
then run some tests that weren't just
Yeah.
based on the protein content and look at whether it actually improved the mixing quality.
So Lee, do you actually have like the genes that control these, or you are just doing?
No, it was not looking at the genes. It was looking at the quantity of what was it, maybe polymerizing proteins, extractable, non-extractable proteins,
Okay.
worked out by, what's his name from USDA? I'm blanking on his name at the moment, but he's our collaborator in quantifying those.
Mhm.
So instead of trying to look at the genes, we were looking at the proteins themselves.
Okay.
I think Xiaofei, when he was there, had tried to look at some genetic control of those things, but
Mhm.
mostly it was just quantifying the
Mhm.
proteins themselves.
Yeah. Cool.
The higher protein that George showed in the Kansas grown TLI line, that that was complemented with the lower starch, which to me points to just a grain filling issue, maybe
Yeah.
with a dry year or just the general dry conditions. Does that land with you, Lee, as a decent explanation, or do you think there's more going on?
Yeah, that wouldn't be surprising. Although, I mean, in wheat, you get your highest proteins not in Kansas, but in North Dakota. So I don't know. I don't know if that's the thing or not, but definitely as part of that breeding for the protein, we're looking at the total protein content as well as the extractable or insoluble, soluble parts of it.
Mm.
So the total quantification from plant to plant, I think it ranged from about like 16% to 30% if you looked at a plant to plant basis. So there's a huge genetic diversity even within our fields here. If it's averaging 20, it's ranging from 15 to 30.
Yeah, and Mitch, if you have an increase in protein, it definitely would impact your carbohydrate and vice versa. So what we've seen with the increase in seed sizes, the carbohydrate is going up, but the protein is gradually going down.
Mm-hmm. Makes sense.
Okay. No more
Thanks,
questions.
Lee. Lee, you're looking pretty good.
Hey, you too, Don.
All right.
All right, thanks very much, George, and
Thanks,
to
George.
all the folks who were part of those studies too. It's a big team, so
Yes,
Great
yeah,
work.
you're welcome. Yeah. All right. Okay.
bye.
Bye.
Thanks, George.